complex anatomy of flowering plants. Following divisions of cells in both primary and as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues and vascular tissues. Type 2. Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissues. Permanent tissues having many different types of cells are called complex tissues. First type of permanent tissue is simple tissues. A simple tissue is made of only one type of cells. The various simple tissues in plants are Parenchyma, Collenchyma and Sclerenchyma. Parenchyma forms the major component within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, secretion. The collenchyma occurs in layers below the epidermis in dicotyledonous plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to a deposition of cellulose, hemicellulose and pectin. Collenchymatous cells may be oval, spherical or polygonal and often contain chloroplasts. These cells assimilate food when they contain chloroplasts. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant such as young stem and petiole of a leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell walls having a few or numerous pits. They are usually dead and without protoplasts. On the basis of variation in form, structure, origin and development, Sclerenchyma may be either fibers or sclerids. The fibers are thick-walled, elongated and pointed cells, generally occurring in groups, in various parts of the plant. The sclerids are spherical, oval or cylindrical, highly thick and dead cells with very narrow cavities, lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear and sapota, seed coats of legumes and leaves of tea. Sclerenchyma provides mechanical support to organs. Now second type of permanent tissue, complex tissues. The complex tissues are made of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Xylem functions as a conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of 4 different kinds of elements, namely, tracts, vessels, xylem fibers and xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracts are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell walls have thickenings which vary in form. In flowering plants, tracts and vessels are the main water-transporting elements. Vessel is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforations in their common walls. The presence of vessels is a characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food materials in the form of starch or fat, and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Primary xylem is of 2 types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem and the later formed primary xylem is called metaxylem. In stems, the protoxylem lies towards the center, pith, and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endarch. In roots, the protoxylem lies towards periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transports food materials, usually from leaves to other parts of the plant. Phloem in angiosperms is composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long, tube-like structures, arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells, which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by pit fields present between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers, bast fibers, are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated, unbranched and have pointed, needle-like apices. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax and hemp are used commercially. The first formed primary phloem consists of narrow sieve tubes and is referred to as protophloem, and the later formed phloem has bigger sieve tubes and is referred to as metaphloem.